Hey guys, today I'm going to be talking about DNA or deoxyribonucleic acid. But specifically, I'm going to be talking about nitrogen bases and nucleotides. So let's just get right into it. Well, okay, so one of the first things we can do is let's ask ourselves, what is deoxyribose? What is deoxyribose? Okay, so deoxy, we can split this definition up into two terms. So deoxy just means to lose an oxygen atom. And then ribose is just our sugar. So this is what a regular ribose molecule looks like. So as you can see here, the only difference between these two molecules is the missing oxygen atom right here. As you guys can see here, we don't have this. We don't have this oxygen atom that is supposed to be here. It's gone. So that's why we call it deoxy, meaning losing an oxygen atom, and then ribose, our sugar. So you might be wondering, why does any of this matter? Well, when we look at a DNA strand, we have to consider the building blocks of this DNA strand. So our building block of a DNA strand is something called a nucleotide. It's called a nucleotide. So we're going to have our different bases coming together here. And this will all make sense in a moment. So I'll do green here. And then let's do red. Red here. No, actually, no, we're not going to do red because that's already the color of our sugar here, ribose. Deoxyribose. So when we're looking at nucleotides, I'm going to make this easier to see. We want to look at a specific part of our DNA here. So remember, it's double helix, so that means it's double stranded. So when we're looking at parts of our DNA, our nucleotide, which is the building block of the DNA, we're going to have three essential parts. Three parts. Oh, let me do that better. Here we go. We're going to have our sugar, our deoxyribose. We're going to have our deoxyribose. Then we're also going to have our phosphate group. And then finally, we're going to have something that I'm going to discuss later on in the video called our nitrogen base. So this is a nucleotide. This is the composition of a nucleotide. And this is the essential building block of DNA, of deoxyribonucleic acid. So this is going to be our sugar. This is going to be our phosphate group. And then finally, this is going to be our nitrogen base. So whenever we're talking about nitrogen bases and nucleotides, we always want to consider four nitrogen bases. They're going to be thymine, adenine, cytosine, and finally guanine. And these all pair with each other together. So let me go more into depth with this idea here. So let's just pretend these are going to be like our holders, you can call them, for our nitrogen bases here. So every time we're talking about DNA specifically, T, or thymine, will always pair with A, adenine. And then cytosine will always pair with guanine. So a helpful way to remember this is T, and A. So T and A, I'll do it actually more like this, A and T. Apples. Wait, that's not the right color. Here we go. Apples in a tree. Apples in a tree. And then for C and G, we can do cars in a garage. So this is a helpful little way to remember what pairs with what. So here I have just a regular DNA strand, and it looks straightened out just to make it a bit easier. And we have our nitrogen bases here. So now we're going to talk a little bit about DNA replication. So DNA replication. Because our DNA has to replicate itself in order for us to grow, right? So DNA replication, it happens when an enzyme called let me just color this in here. An enzyme, wait, that's not a very good draw. Here, let me do that. An enzyme called helicase comes in and it unzips our DNA, right? So this is going to be helicase, and you can just picture it kind of like as a zipper, kind of like unzipping our DNA here. And then right behind our helicase comes primase and polymerase. And what these guys do, so this is going to be our primase, and then this is going to be our polymerase. Let's do blue, dark blue. And the polymerase is going to be helping build a second strand of DNA here, because it has to replicate itself. So all of our previous nitrogen bases that were up here, so yellow, pink, they're all going to be copied, and basically like a new strand is going to be formed. So what's going to happen up here is yellow's going to eventually connect with blue, pink is going to connect with purple, and so forth and so forth. So, and then down here are remaining strands that survived. Here, let me do a different color. Let's do green to show you guys here. These strands, because this was basically cut in half, these guys are going to come down here because our helicase is unzipping our DNA. So, wait, no, that's got to be purple. So basically, it splits in half, but as it's being split in half, a new strand is being made. So as you can see, as you guys can notice here, this base and this base are the same because they were the same here in this strand. They were the same here in this strand. So once we create our two copies here, what's going to happen is this is going to become a totally new strand, just like the one up here. And then down here, we're also going to have a new strand because our primase and our polymerase is also going to be working upon here. So here we're just going to add our yellow base, our pink base, and then another yellow base. Okay, so our DNA has now been split up by our enzymes. So now, what do we do with the DNA now? Does it just remain unzipped? Well, no, it doesn't. Another enzyme called ligase comes in, and it travels along the DNA to help close it up. So when the DNA closes up, we officially have our sealed up DNA strand, right? So that is basically DNA replication, and what will actually happen in the end is we will have two DNA strands, and that's basically how DNA replicates itself, and then it eventually prepares itself for mitosis in somatic cells to be able to divide and create more cells and grow. We'll go into that in the future. I'll explain mitosis and meiosis, but I just want to start here so that you guys can have an understanding about DNA and how it replicates itself. Thank you for watching. I really hope you guys enjoyed, and let me know if you guys if you guys want to recommend anything, and then I'll try to cover it. But yeah, if you like this kind of content and like to see more science and math videos, make sure to subscribe, turn post notifications on so that you never miss a video, and also like the video up so that other people can see it. Thank you so much.